Please help me welcome Uwe Bernstein.
Thank you very much, Scott. Yeah, it's a great pleasure to finally be here. It was quite a day. So I may not be the best performer tonight, and I hope that those of you that came here to have bonus points on their grades feel that's going to be worth it. Okay, so it's always tricky to plan a public talk. Try to adjust it to make it of interest to people that have a biology background and perhaps people that don't have a biology background. So we'll see how that's going to go. So basically, the talk will be about using DNA for surveying biodiversity. So we may as well just start the talk talking about biodiversity, and by taking this quote of Edward E.O. Wilson, who actually was the first one to coin the word biodiversity and put things into context about where we are with our species diversity. So Edward said, species extinction rate caused by human activities is 10,000 times faster than during the 5 preceding geological episodes. So there's been this report that came out this year from the Global Assessment Report on Biodiversity and Ecosystem Services that makes a very exhaustive, comprehensive update on where we are now in terms of the situation with our biodiversity. So this first figure illustrates the cumulative percentage of extinct species across vertebrates groups, and you can see that quite over the last, since the 20th century with the industrial era, especially for amphibians and fishes, extinction rate have been ramping quite quickly. In this report, we find also quite a few tables that report on the proportion of endangered species. This table is for vertebrates, again, that are threatened with extinction. You can see that the percentage, especially for amphibians, and skate and raise this percentage of threatened species across the globe is pretty alarming. And just last week, this paper came out as well, just to illustrate that the biodiversity crisis does not hit only the big animals, only the vertebrates, but also the insects. And so this is a work that has been done in Germany. I'm not going to go through the figures, but it shows that over the last 10 years, just about in either grasslands and forest area, the overall biomass of insects went down by 2/3, the abundance by 80%, and the species number reduced by 1/3. And similar numbers in the forest, except that the abundance of species remain even over the last 10 years. So that's pretty dramatic as well. Then we're also losing diversity at the intra-specific level, so that's what is illustrated on this slide here. It's the cumulative number, it's the reduction in the number of populations, again, in various vertebrate groups, in terrestrial species, marine species, and freshwater species. And basically, overall, the estimate is that the actual number of population for the species that were surveyed has been reduced by 40% on average over a period of 30 years only. And as you may imagine, in terms of absolute numbers of species that are threatened, those mainly occur in the tropics, and it's illustrated either in marine environment with the dark blue or the dark green on the continent. It illustrates, so basically the darker the colors, the more absolute number of threatened species you have in various areas. So you may think that we're doing quite good here in Canada, but when you look at the numbers, compiled by the COSEWIC. Here in Canada, we have actually over 771 wildlife species in various risk categories. So we have over 100 species that are endangered, 183 that are threatened, a couple hundreds that are of special concern, and we also have extirpated and even extinct species. So we have problems as well. So, and then the reduction in biodiversity tended to hit actually the larger species, and the report I was referring to shows that this is true for mammals as well across the globe. And here, that's an illustration how related to overfishing exploitation, how we have been fishing down the trophic chain, if you want, and first overfishing the larger species, and now we're basically targeting the smaller fishes around the globe. And even within species, we have been overfishing fish with the best genetics for growth potential, like in cod, for example, such that this type of cod with a high genetic growth potential, we may never see them again because we over-harvested fish with the best genetics for growth in cod and other species as well. So all of that is not going to improve unless we start to do things differently. So over the last 50 years, we really have transformed our environment more than during our entire history. So those are several of the threats that organisms are facing. Obviously, change in temperature related to the greenhouse gas, in the marine environment, ocean acidification, but also habitat alteration, forest burning, the plastic world, poaching, etc. So not too good. And this has consequences related to the degradation of many ecological services that are rendered by ecosystems that we are impacting. So these figures, I'll just pick one of these figures. So the first one relates to the water quality. And what you have here again, it's illustrated by a gradient of colors, and the darker the green, the higher is the nature contribution to help to solve our problem. In this case, water quality regulation in terms of the proportion of pollution avoided by nature contribution. So major ecological services rendered by the ecosystem and the organisms that lives in it. So this trend is on the increase in the 21st century, as you know, and in terms of subsisting, us humans will be forced to increase the pressure on our environment, which may worsen the loss of biodiversity unless we seriously think about doing things differently. So this is why perhaps it's the time to listen to the young voices of reason. Okay, good news. I'm done with the bad news. Was just to put things into perspective. Well, good news is that there are so many species that are still to be discovered and identified. So actually, scientists estimate that the number of species to be anywhere between 8 and 30 million. It's very hard to be precise on that. And so far, we have identified only 2 million of those. Of course, most of these unknown species are part of what we call the neglected biodiversity. Mainly invertebrates, algae, fungi. Those represent over 95% of the biodiversity, and probably more so of the unknown biodiversity. So currently, about 16,000 new species are described every year. So at this rate, using traditional methods, it may take between 500 and 1,000 years for scientists to complete the species inventory. And there are very many reasons for that. First, identifying each specimen of each species using traditional methods is pretty cumbersome. For example, you have to become very, very expert in using taxonomic keys. It takes time. having the manpower to do that also is a problem. And the training of taxonomic specialists is not really on the rise these days. It's hard to get systematists hired in our departments these days. So other problem also is that there are lots of species that look very alike. So those are not representative butterflies of the same species. They are all different species, and same thing for these frogs. So unless you are very, very expert, you may have a very hard time to identify these species, except like investing a lot of time and energy. So we need methods to boost our capacity to identify and monitor species faster. And Paul Hebert from University of Guelph put that things, the complexity of biodiversity, put it into perspective. I like this quote of Paul saying that whereas physicists deal with a cosmos assembled from 10 fundamental particles, biologists confront a planet populated by millions of species, which is true. So we should not be ashamed to be biologists, right? We face very complex problems, as much as astrophysicists. So that led Paul to propose the concept of DNA barcoding back in 2003. So basically, as a diagnostic species identification genetic barcodes to rapidly and efficiently identify species based on genetic variation in DNA. So basically, DNA barcoding is defined as a method allowing to identify species from a short sequence of DNA. And that short sequence of DNA, there's variation to that, but basically it is a small pieces. This is the cytochrome oxidase gene within the circular mitochondrial DNA that is present in every mitochondria, which is the energy power in our cells. And this gene is present in all animals, to speak specifically about animals, such that you can use that as a standard that look at genetic variation between species in that short stretch of DNA, which is very easy to analyze and very cheap also, and in this way to have a diagnostic identification of specimens. So how the DNA barcoding initiative works, basically, so you need to, the basic idea first is to build a reference database, and then you can use this reference database that is currently being built to identify unknown sequences and see what species it refers to. So to build a reference database, of course, you need experts at the beginning to identify specimens, and those will be, if you want, holotype specimens and put in collection data, for example, in museums and so on. Photographs are being taken as much as possible. A small biopsy of any piece without destructing the whole specimen is being taken to do the DNA extraction, which will be sequenced for this little piece here, and this will be the genetic information that will be used to identify different species. Put a reference sequence to a reference specimen. And all of that is being centralized on servers at University of Guelph. So the point of this whole diagram here is just to show that at the very end here, all these databases are available not only to scientists but also to any possible users, and also to the general public for as an encyclopedia of life. So has been a contentious initiative, because people were saying that you cannot really have much confidence to put diagnostic identification on species on just sequencing a little bit of DNA. But overall, I think it's a successful, very successful initiative worldwide. because it's standardized and fast. It does not require a deep taxonomic knowledge of everyone being involved in the process. So you need experts, but it's not the experts that's going to be involved in identifying each specimen, because they will be involved in building the reference database. But then afterwards, for the sequencing and stuff, you don't need to be an expert in the actual taxonomic groups to put a species name on your sequences. And the online database is there and free for everyone. So right now, Paul Hebert from University of Guelph, I insist on him because this is an initiative that has been that was born in Canada, and the driver of this major worldwide initiative is here in Canada, University of Guelph. So right now, there's that $180 million initiatives with the goal of discovering 2 million new species all over the planet in the coming years. Okay, so then coming to some of the work I've done in the past in link with the barcoding initiative, I was responsible for building the reference database for all freshwater fishes in North America, and this was published in PNAS a few years ago. So essentially what we did, we sequenced nearly 6,000 specimens representing 752 species that were identified by us or other experts that could actually identify the fish. And the representative specimens could be conserved in collections for 80% of the 752 species. So to make a long story short, within those 752 species analyzed, we identify over 200 what we considered new candidate species, in the sense that that morphologically those species were, or these population that were, well, things that were considered population within species were from the morphological similarity were considered population within species. When you look at the DNA level, there were lineages within species that were highly divergent for the rest of what was considered the same species. And this is how we, there's like basic standards of level of divergence that are being more or less accepted as past that threshold, you can consider that you may be in presence of new species that have been unnamed or undetected before. So 200 of those. So this suggests a potential increase of nearly 30% in fish biodiversity in North America. So we were not happy enough to publish in PNAS this work, so we just went up and published in the famous Journal of Montreal afterwards, using DNA barcodes as well, but in a quite different context. It was one of the very first studies we've seen quite a few of those coming that have been come out in newspapers over the last few years. So this was done 8 years ago, and basically I work with those newspapers and we collect Quebec in restaurants and grocery store to see if what you were buying was what you were eating. And obviously I will not present that if it was like 100% of what we bought is what it is. No. So basically overall about 50%, so those are the sum of all samples being analyzed. So basically 50% of the samples we analyzed had the wrong identification, either on the menu in restaurants or in grocery stores. And you have some illustration here of some of the classical misidentification. Vivano is forgot the name in English. It's a snapper, red snapper, systematically replaced by tilapia. That's a classic case. The bluefin tuna, which is red tuna in French, this is more, I would say, like not necessarily a bad intent, but it's really a labeling problem because the flesh is red. In the fish market they will call it red tuna. They will call it red tuna, which is a bit silly. But the Canadian the Canadian agency, Canadian Food Agency anyway has a website, so it's easy to understand also why partly things are mislabeled, because the nomenclature that is accepted in terms of vernacular names is a bit of a mess. But there's also bad intents behind that. So in that article, we even published, that's in Montreal, the name of the restaurants and what you could eat there instead of what you had ordered. Like, for example, lots of tilapia, and so yeah, so those are restaurants that probably not the best place to eat in Montreal. So this is still relevant today. This came out again. That was another similar study of that type done by this group Oceana across Canada, and so that was published. That came out 2 or 3 weeks ago, no more than that. So things has not changed over the last 8 years. 47% of the fish tested in Canadian retailers were mislabeled. So just about the same proportion that we have seen across the province of Quebec. So clearly, something should be done to do things better in terms of traceability, and DNA barcoding could be a very, very easy way to do it, but government don't really do it yet. So they should do it, and it's cheap. Okay, so moving to DNA barcoding being done on actual tissues from collected specimens, bring us to the world of environmental DNA, which is basically using the DNA barcoding concept, but applied on analyzing DNA not taken from tissue samples from the animals, but DNA taken in the actual environment, DNA that are released by organisms by various processes, just skin shedding or whatever you may think, feces, etc., release DNA in the environment, and that DNA, depending on the conditions, can persist for a very long time. So we can actually go back in time and get DNA from past environments and look back in time what was there in terms of species and so on in the past, and of course comparing with the present. So Scott mentioned that until recently there was no specialized journal, even if like this whole world of this whole field of eDNA is really exploding these days, and I just realized that there was no journal, so I said, okay, I'm just gonna start a journal, and so this was launched in October 2018, just to say how young the field is, but also how expanding it is, and it's not gonna stop. There's just so many things that can be done with that. Okay, so in the very broad sense, there are two main things that we can do. So environmental DNA first can be defined as a method allowing the detection of species from DNA barcodes. So mainly the vast majority of environmental DNA work being done is done on the same stretch of mitochondrial DNA or other genes in the mitochondrial DNA, but it's basically applying the principle of DNA barcodes, but on DNA found freely in the environment. So there's two major approaches that people use. You can, depending on your protocol, you can either apply a species-specific approach, so you're mainly interested in the presence, absence of a particular species, or also having some estimates of the amount of DNA of that species, or you can also apply a community approach, let's say for a given group of organisms, let's say fishes or salamanders or marine mammals, for example. If you stay in the aquatic environments, then you can have what we call universal primers that will allow you to detect the DNA of all species of a given taxonomic group you're interested in, for example here in fish community. So I'm just going to show you a few examples of recent application of eDNA, just to show you the potential that these methods offer. So that's a very recent study that was published this year in Science Advances by a group of French researchers. Work being done in the New Caledonia, in the Indian Ocean. And basically the researchers here were interested to compare the efficiency of the community approach, eDNA community approach, and compare what they would detect in terms of shark diversity compare with traditional methods that have been used previously in the same system. So basically what you have on this map is just a bunch of locations in greens and blue that were surveyed by visual inspections and other direct visualization methods, and those in reds are locations where people took samples of eDNA and see how many species of sharks they could detect in the system. And those are the results. So basically, in green and blue are the two different conventional methods, and you can see that both methods could identify 9 species of sharks, but with lots of efforts, hundreds of observations being made with this method. The one in the green, thousands of observations, whereas with 22 samples, water samples, and getting the eDNA out of this couple liters of water. You filter the water, the DNA gets stuck on a filter, you extract DNA from that, and you sequence and you see what species you have. And they have identified 13 species as opposed to 9 species, even doing only 9 locations. So clearly, and many species, these 6 species were never detected by the other 2 methods, even after years of inventories. Okay, so closer to us, this is a work I've done with the Department of Fisheries and Oceans, again using this eDNA community approach. And here the idea was to compare the eDNA efficiency with the trawling method that Fisheries and Oceans do annually in the Gulf of St. Lawrence to document demersal fish community. So they were interested to compare to see what they were seeing in terms of species diversity in the bottom trawl compared to what we would see with the bottom water samples, and see also if we could get some semi-quantitative information also in terms of relative proportion of biomass of different species. So don't bother with the different colors, doesn't matter here. So we had 85 sampling stations, and of course we took water samples at the bottom and obviously before the trawls. So that's the comparison of what have been seen. So eDNA detected 71 species, the trawl 64 species, and only 64% were shared between the two methods. So clearly, it's for me, it's worth basically use things, not to say that one method is better than the other, but depending on your questions, use different methods in complement is probably a good way of seeing things, which means that people should think about integrating the eDNA analysis as part of these surveys, at least. It was interesting also to see that the most abundant species were pretty much the same detected by the trawls and the eDNA. By far the most important species in terms of biomass for the trawl, or in terms of the number of sequence reads that we had in our analysis, was redfish, sebastis, and proportions were quite comparable. And then in terms of the ranking, screening, like the second, third, and fourth species, the capelin, the Greenland halibut, and the herring were the same for the trawl and the eDNA. The big difference was that, like all the other species, represented only 4% of the biomass in the trawl, but represented 22% of all the sequences we had in this study. But there was something comparable, at least for the most abundant species. Another example of an application that we have done recently in a totally different context, but still in the marine environment, was working with Parks Canada as part of their annual surveys for cetaceans, for whales, fish prey in the St. Lawrence River estuary. Actually, this is a provincial national marine park, one of the very few that we have in Canada, and it's a major area, a feeding area for whales. And this is because there's a major upwelling area in this upper part of the marine estuary, and the cold Labrador current flowing at the bottom, coming from the Arctic, very rich in nutrients, hit this wall here in the estuary, creating this major upwelling zone. And every year, basically, 10 different species of whales are being reported in this small area. So that's a very amazing area. So Parks Canada are interested to know, for those species that mainly feed on fish, what species they feed on, and just try to track the abundance of the different species that the whales are feeding on. And they have been doing that by using hydroacoustic. But so hydroacoustic can be very helpful to detect fish schools, can be useful to guess what species this based on the frequencies that have the biggest hit. You can make some guess of for example, if the fish or those are fish with a swim bladder or not. But there's limitation in terms of identifying species that you have composing the fish school based on hydroacoustic. So Parks Canada were interested to see if we could use eDNA to validate what they were seeing by hydroacoustic. So the way we were doing that was really a lot of fun. So we're going out and then by hydroacoustic locating a fish school like this here. So sometimes fish schools can be very huge. I never realized that. So this thing, like we found capelin schools that were 3 kilometers long. I never knew that was possible, and 6 meters thick. That's crazy. So anyway, that's one of those schools. So we're locating schools. Then we stay still, and we suppose that fish moves, right? They should move, school. And we take and we drop like Neskin bottles down within the school. So we take few samples, 4 or 5 samples, and just from there identify the species that the most likely, the species that comprise the schools. Here we use the species-specific approach, but for 4 different species that are the main known, the most likely forage fish in this area. So redfish, capelin, herring, and sandlace. So that's a couple examples of how we could identify the fish that were composing the school. So you have two schools here, and basically what you have is the little graph shows the amount of DNA, or in terms of number of molecules detected for the 4 species we were screening, and then you can see that basically these schools were redfish. redfish schools. And what I did not mention is that we run negatives also with that is taking water samples out of the school, because obviously there will be fish DNA hanging around outside the schools, and we subtract that signal, we subtract that amount of DNA that we see in negative from samples within the school to establish what's the real signal. For example, these things here would disappear when we subtract what was being seen in the negatives, confirming that this is those are redfish schools. Whereas in this case here was a capelin school, and here we have a mixed school composed of herring and capelin. So that's very, very useful, and that allowed them to really validate and complement the work that they've been doing with hydroacoustic. Okay, other things that environmental DNA have been very useful for has been to track the invasion of new species. And one famous, or I'd say group of famous invasive fish species that have been tracked in North America is the so-called Asian carps, or Asian carps invasion. So basically, there's 4 species of those, and basically it all started when these 4 species were imported from China for plant control and in fish ponds and other control, but quite quickly the fish escaped the ponds. Every biologist would have predicted that, and just started expanding throughout the Mississippi system. And right now the big fear, especially for this thing like the bighead carp and also the silver carp, is that these species can eventually cross from the Mississippi drainage into the Great Lakes system, because there's only the Chicago Canal. There's a Chicago Canal that connects the Mississippi drainage with Southern Lake Michigan, and the fish are only kept from moving from one system to the other by electric field. And sounds like something may happen. And just to show you, and that's pretty bad. Probably you've seen this thing, but it's interesting. It's always spectacular to see. So system where two of these species, the bighead and the silver carp, invade, like in the upper Illinois River. That's the density that they can reach, and basically they can represent like over 95% of the overall fish biomass in the system that they invade. So no wonder we are really scared that they would invade the Great Lakes and the St. Lawrence River system that supports besides supporting a very important local biodiversity, support fishery industry, both commercial and angling, of over $7 billion. So we're not sure we want those fish to make it to the system. So there's a lot of effort to monitor the presence of these species, among other things using eDNA. So I did some work with the provincial government in Quebec, and because obviously they were worried about the possible presence of some of these one or more of these 4 Asian carp species in the system. So we developed the assay to survey them through eDNA in the system, and we did that over 2 years, 2015 and 2016, and we detected 16 positive locations, which are in red in the system here. So I failed on my story. I should have said earlier that this work was undertaken after this commercial fisherman caught this 64 pounds grass carp in 2015, and the government was obviously worried and wanted to know if there was more than that one carp in the system, and that's how we initiated this work, and ended up finding DNA of this grass carp species, all the way from Montreal to Lac Saint-Pierre, which is about halfway between Montreal and Quebec City. Closer to you, smallmouth bass invasion. So work of Antoine and Alan, and I've been involved in running eDNA with them on the smallmouth bass, following the observation that smallmouth bass were been found in Miramichi Lake, and Alan and Antoine quickly thought that eDNA could be useful to see if in the early days when they were reported, if the smallmouth bass could have made it its way all the way down to the Miramichi River. And this is what came out of this first analysis. The green spots identify positive detection of bass DNA, and you can see here that bass DNA was detected in some location in the Miramichi, and Alan actually eventually visually spotted a bass in the system. And of course, since then, more observations have been reported, and this this was published recently, just reporting that now over 30 observations have been made within the Miramichi. But the eDNA analysis allowed a very early detection, probably before the fish was more present than that in the system. Okay, and last example, and not the least, we'll see how far you can push it. We're not looking for the existence of Nessie in Loch Ness, and that was, who have heard about that story? Raise your hand. 1, 2, yeah, that's a cool story. So Neil's a pretty creative guy. So he's from New Zealand, and obviously, I mean, he never thought that he would discover the DNA of Nessie, but it's basically, I guess, what he had in mind was just to popularize science more than anything else, and they thought it would make a nice public scientific splash with that, and it did, because it became famous all over the world for that study. Yeah, so basically, but he did a pretty extensive survey in the Loch Ness, hundreds and hundreds of samples, and he used the community approach not only for fish or vertebrates, but also invertebrates. So he actually did a very, very beautiful ecosystem approach using the community approach of eDNA. And of course, so that created a lot of frenzy in the popular press, and with a little bit of science bashing. The search for Nessie collides with some cold, hard science. Anyway, but he didn't plan to say, well, you know, there's no monster, this and that. So he found a way to keep the legend alive, but they changed the story a bit, and saying that there's a lot of eel DNA in Loch Ness, and some eels have been reported that have been seen in Loch Ness to be, I don't know if they were giant, but very, very big eel. So he just retained the idea, he pushed the idea that perhaps Nessie is actually a giant eel, but that was taken in the press as well. But no monster or dinosaur, but maybe a giant eel. Anyway, to conclude, I guess biodiversity is assaulted on many fronts. Animal and plant species are declining at an alarming rate, yet millions of species are still to be discovered and described before they actually vanish. But unfortunately, traditional inventory methods and the manpower necessary to do it cannot keep pace with the effort needed to identify species using traditional methods. So DNA analysis, both DNA barcoding but also environmental DNA, are very powerful tools that can help and which cannot be ignored any further for management and conservation of biodiversity. And on this, I thank you for your attention.